In terms of the first topic that we're going to talk about today, it has to do with file formats, data formats. I just also want to put another plug in for the GitHub page. So the SMS Colab page, and it is another resource in addition to the website, linked from the website, that includes all of the resources that we go through and present. So you can look actually at all of last year's lectures and resources. Again, we've changed the course, incorporated spatial. We've added new lectures from different lectures on single molecule course, so it's definitely not identical. But if you are interested in more sort of resources and practice in the way that we've formatted it, or if you like the format that we've been presenting, there's more content for you to check out there. And in terms of, so you can see for course one, we have the slides from last time and homework posted, and after Friday, the key will also be posted in the same place. So I'm going to take the link to L2 here. If you want to follow along in your own Colab notebook, I'm going to put that in the chat for everybody and then present it here on screen. So we have sort of a broad range, familiarity, differences with Python in the audience. So I'm going to start from a broad-based tutorial. In terms of, the Colab notebook, its purpose, the environment, it's basically a product by Google that can provide free computational resources, including access to GPU resources, which is necessary for the base calling algorithm that's run within this program. In terms of the data and data management, there are sort of multiple ways of lots of different ways that data gets managed. What I've done here is mainly post it on Zenodo, and then you can download the raw data from Zenodo with this wget command. And once you do that, it places it here into the files box. Zenodo is good for this kind of subset data. It's good for anything in essence that is under 50 gigabytes, which is sort of the maximum file size that you can store within a particular window, box, or setting. And if you go, you can actually find this on the internet where I've posted it. And eventually, as we get to your final projects, there's actually a data page that's associated with this course that will be updated with all of the data sources and published data links that you can use and download into your own environments. So that is the download data piece and element. So if I could just run that here as an example, it very helpfully and nicely started up a whole computational server for us. So just to get a read of the room, if you could all answer in the chat, so open up the chat and answer for me how many of you have worked with BioConda before? It's just like a yes, no answer. Okay, and if you have worked with BioConda before, can you put down your favorite package? This command here, it installs this is actually a series of commands. So this exclamation point before the command means that it's a shell command, um, versus if there's no exclamation point in front of it, that means it's Python syntax. Okay, PySam, that's pretty cool. We're using SAM tools here as well. So if you do look at my code, what you might notice is that this is a really, really old version of BioConda 4.8.2, which happens to be compatible with Google Colab. If you do decide you wanted to install the newest version, I think it's 37 point something. But yeah, if it's not broke, why fix it? Goes the mantra. But yeah, so this is a little bit of an older version. You also see that this is called miniconda. Miniconda versus BioConda. Miniconda is basically a smaller packaged version, so it's less storage space overall. It doesn't include any sort of pre-built packages. So if you decide to go to the Anaconda website and you download their regular Python installation, you'll see that it's got a lot of nice, helpful data science libraries built in for you, such as NumPy, SciPy, Panda, all those nice things are sort of pre-packaged for you to use and to work with. Whereas miniconda excludes all of that and just assumes that whatever you want, you're going to build yourself. Is anyone familiar with why BioConda is so important for package management in particular? In essence, right, what I am getting at with the question is that if you have something like SAM tools, maybe, and if you have another program Y, and they both require pandas, this other library, because they both like working with data frames, they both like working with these sort of spreadsheet-like data formats, and they both access different kinds of commands that are available in pandas. If you if you have these two packages, you might think, oh, I'm just going to install pandas, I'm going to install SAMtools, I'm going to install package Y, happy days, right? But let's say that those two packages were actually developed at different times, and as a result, they require different versions of pandas. How are you basically going to how are you going to sort of manage your computer, your software systems to make sure that when you're using SAMP tools, it links to the pandas version that it needs, and when you use program Y, that it links to the pandas version that you need. So at a certain point, right, you need two pandas versions because I said they require two different versions. They were developed at different times. And you need to sort of containerize it in a way that the program knows on your computer which pandas version to use, and it doesn't mess up. That's really the real part of what Conda is and what a Conda is about. It's this package manager that lets you separate out into different programs into different environments and install their dependencies independently of each other. And so that's very important for understanding, um, basically making sure, right, that you're able to run each piece program within a pipeline, even if they don't share all of the same dependencies. And ultimately, right, when people talk about transferring software, they're both transferring, right, a piece of code and a code base, but also all of the versioning for the dependencies that they might be working with. So the reason why we've installed it here, I don't believe that this code creates any individual environments or like separates all of the programs out because all of their dependencies, again, are compatible with each other. So there won't be a conflict. But that is sort of the major innovation and the beauty for having something like miniconda, which abstracts a lot of the details of that dependence management away from the programmer or the user, lets you focus on what you actually want to focus on, which in our case is the data. So let's just do another yes/no question. How many people have heard of FAST5 files? Yes, no. Well, that's true. That's true. If you look at the history of Nanopore file formats, they have a history of regularly changing them. I think that's something that their customer base really appreciates, and that's why they're very loyal. But change it. They've changed them a lot, and so at the beginning they were, in a sense, less usable, and then they became more and more usable, or considerably more and more usable over time. Um, But to go back to the Fast Five, so I put some notes on the screen as to what these file formats are, and they're everything that people have started saying about them. Something that comes off the Promethean, something that also is flexible enough to store and data or single cell at its core, and something that's a precursor to the FASTQ. And so to go into a little bit more depth, to understand what the Fast Five is. It's a far more multi-dimensional, or what I would call multi-dimensional object than a fast queue. So whereas and it probably includes somewhere between 10 to 100 times more data than an individual fast queue file. And so the Fast Five needs to sort of be much more expanded, needs to be in a format where it can handle all of this extra high dimensional data that gets abstracted away when you move into the FASTQ file. So if you look at, for example, here I have HDF5 tools. So HDF5 is basically the file format that FAST5 is built on. So it's this sort of shared data format, data structure, where you have groups, data sets, and attributes, with the groups and data sets being these multidimensional arrays of data and the attributes providing lots of metadata, and you being able to flexibly customize how many attributes, how much metadata, and then what the input data is. So yeah, and it's similar to JSON format, if anybody is familiar with that from public queries. What I've done here with this h5ls command is just to sort of print out some example indices for a FAST5 file. So this read here, and with this unique ID code, represents a single read number. And then these here are the different locations of the different signal. So the signal data set being the actual raw electrical signal that you get as a piece of DNA or RNA moves through a nanopore, and then the remaining attributes here. And what I was alluding to at the beginning, or what I tried to, I guess, make light about, was that when Nanopore first started, they output everything as single read FAST5 files. So let's say you did an experiment with 1 million reads. You would get 1 million different files, each with one, and each FAST5 would have a single read, and it would only containerize a single read. And then eventually they figured out that people didn't want millions of files on their hard disk to manage. And so they merged it right into this other format, which is the multi-read FAST5 that contains all of them within this kind of format. And then you can use an HDF5 library in order to take a particular read, as I've done here, and output its group, its different attributes, and the different data values that are associated with those different attributes. So yeah, you can see here the signal, start time, start mux, read number, all of that. So I've also put here a little note on what these signals mean. So signals are much more high dimensional, as I mentioned, than a nucleotide, a sequence of nucleotides or reads. And it turns out that these were originally measured as picoamps, stored as 16 integer values, but then ultimately transformed for the sake of their reporting here in this sample piece. So ultimately, the comparable unit, or the rawest unit of the signal, is its original picoamp value. And if you look into their APIs and you look into their documentation, you can see that by taking the data that is available within the actual raw signal format, you can convert and get back to that original picoamp value for downstream comparison and analysis. So that is the FAST5 format. Nanopore has recently released and is switching to a new format called POD5. This has been around a year. It relies on Apache Arrow, and the main innovations have to do, instead of sorting it as a JSON file or like in objects and metadata and such, this is a column database storage system, which works much faster. If you look at it, that means within the ecosystem, what you're learning about today is that in terms of nanopore signals, you can store things as FAST5, although now it's being deprecated because it's been innovated on by POD5. You can use POD5, which is now the newer and faster format from Nanopore, faster for querying, faster for the it's something that Nanopore, the base caller, is optimized to work with, and Blow5. And you'll hear from the developer of Blow5 later and what the advantages of that system are. And he also developed slow-5 tools, which can convert between blow-5 and fast-5. And their group also developed the blue crab tool for converting between blow-5 and POD5, or their collaborator did. So you can see that, right, the ecosystem is very tight and interconnected. And at the end of the day, they're different ways of representing all of the same kinds of information, with fast5 being the first data format that was developed, blow-5 being the second, and POD5 being the third. So this is a demonstration of the POD5 format and its power. I do go through in this example, right, just some use cases of the POD5 library. So for example, I put a 1.46 gigabyte example, a POD5 file up for you guys to use. And even at 1.46 gigabytes, it's only 12,000 reads, 12,000 signals, just pretty astonishing. It also, can output the data, take the data that's in the tabular format and output it as, this more table-like structure. And what you can start to think about and appreciate in any data format is that it's very important to understand the associated metadata. So I don't know how many people have worked with POD5 or otherwise, but when I encounter something that I'm not familiar with, whether with this or with anything else, I start now just sort of teaching myself with ChatGPT and working through different components and different pieces. What does this column mean? What is this metadata? What is that? And so the first thing that I looked at was, for example, the end reason. So the end reason speculatively, and then research confirmed that this refers to when a DNA or RNA is working its way through a pore, what is the reason why that DNA or that RNA ultimately leaves or exits the pore? So there's actually 4 different possibilities for what you can report out. And those possibilities, so signal positive refers to a positive outcome. The DNA or RNA was assumed to move through the entire pore without issue and made it through the other side. Signal negative means that there was something, the signal was not recognized, there's something funky about it, it didn't terminate as you would expect. Mux changed means that basically, actually, I'm going to explain that one later. It requires a little bit more context, but unblock mux change basically means that the molecule, whether the DNA or the RNA, for some reason blocked the pore. And the pore has this cool voltage gating feature that allows it to eject the molecules, right? And try and send it back basically in order to free up the pore and capture another molecule. And so this signal means that that particular signal or whatever was associated with it was taken back. So what does this mux change mean and how do we begin to understand it in the context of these data formats? So if you look at a membrane, a pore, what you'll see is that there are lots and lots of nanopores, so these proteins that you can take DNA and RNA and scope through. And those pores can actually be thought of as existing in groupings of 4. So every 4 pores can basically be taken together as one. So for example, I could take this red one, this green one, this yellow one, this purple one, group it. That's 4 pores together. And each group of 4 pores is considered a channel. So if you look here at the 4th column and you think channel, every group of 4 pores is equivalent to a channel. And then basically what the nanopore does is that it tests out those 4 pores and it decides which of those 4 is going to work the best for sequencing at the moment, right? Which one is sort of the freshest, not damaged, doesn't give wrong signals. It selects that one and basically you start sequencing and within each group of 4, you're only using one pore. And within each channel, basically, you can only be measuring one of the pores at the same time. And so every 1.5 hours, depending, or 8 hours, depending on how you set it, you're going to do something called a mux change. And that means that every read that exists within this lawn, within all of the, within all the channels. So for the one nanopore that's, sequencing at that present time, every 8 hours, all of those pores will stop. They will eject all of the reads back out, similar to like the mux unblock, but that's why it's just called a mux change because there wasn't anything wrong with the reads. You've just decided you're going to unblock everything. Yeah, so you've just decided that you're going to change everything. And it basically picks a new pore within the mux. So it picks one of the other of the four pores, and then it sequences with that for an hour and a half, for 8 hours. And ultimately, the reason why is that it wants to basically refresh the pores and give you higher quality sequencing output overall. And then if you look at, right, the flow cell degradation over time, it's much better with sort of this like comprehensive like pore output and switching versus the other thing. There's also a video here from Nanopore looking at it. I don't need to get into that particular plot now. But what I'm saying is that by examining, right, this particular metadata, understanding what each column means, you begin to actually understand much better what is happening with this particular technology. And you begin to understand all of the different ways that you are able to potentially look at quality metrics and scoring. So how's the quality of all of the signals from the third mux from all of the different channels versus the fourth mux? Or understanding whether there are channels that are systematically, for some reason, damaged within your sample across all of the read times and cycles, or whether there's certain channels whose read quality degrades much faster. Those are the kinds of questions that you can start asking in examining all of that particular metadata. So I have managed to go through the first pieces. I know that flow 5, I included my notes up from last year into the notebook, which you are free to review after the course with the homeworks and however else is helpful. FastQ. How many people have exposure to FastQ files? If you want to put a little yes, no in the chat. Okay, a few people. Um, so for those who don't, so a FastQ file is a text file. So what I should have also discussed a bit is the difference between binary files versus plain text files. A plain text file is something that you can open up. A plain text is basically something that you can open with a Word document and you can read. Basically, it's human interpretable. The data is right away. Whereas a binary file basically is basically compressed in such a way that you save storage space, right? It's easier to store on the computer and it's optimized for computer storage. But if you looked at it in a text editor, you wouldn't understand what's going on. It's like it's almost encrypted, but it's not encrypted so that you can't see it. It's really just encrypted to optimize on storage space. POD5, Fast5 or at least POD5 is typically binary formats, whereas FastQ is a plain text format. The difference between BLO5 and SLO5 is that SLO5 is a text-based format. BLO5 is a binary-based format. SAM versus BAM. SAM is a plain text format. BAM is a binary-based format. So, but the FastQ plain text format is basically this one that's shown. Each read is just 4 lines. The identifier, the actual sequence, some random third row, which actually doesn't have a defined nomenclature, so you can make that third row whatever you like. Some people leave it blank. And then the quality scores in the fourth row, which are the phi-read quality scores.